taste. Gustatory perception, or gustation, is the sensory impression of food or other substances on the tongue and is one of the 5 traditional senses. Taste is the sensation produced when a substance in the mouth reacts chemically with taste receptor cells located on taste buds. Taste, along with smell and trigeminal nerve stimulation, determines flavors of food or other substances. Humans have taste receptors on taste buds and other areas including the upper surface of the tongue and the epiglottis. The tongue is covered with thousands of small bumps called papillae, which are visible to the naked eye. Within each papilla are hundreds of taste buds. The exception to this is the filiform papillae that do not contain taste buds. There are between 2,000 and 5,000 taste buds that are located on the back and front of the tongue. Others are located on the roof, sides and back of the mouth, and in the throat. Each taste bud contains 50 to 100 taste receptor cells. The sensation of taste can be categorized into 5 basic tastes: sweetness, soreness, saltiness, bitterness, and umami. Taste buds are able to differentiate among different tastes through detecting interaction with different molecules or ions. Sweet, umami, and bitter tastes are triggered by the binding of molecules to G-protein-coupled receptors on the cell membranes of taste buds. Saltiness and soreness are perceived when alkali metal or hydrogen ions enter taste buds, respectively. The basic tastes contribute only partially to the sensation and flavor of food in the mouth. Other factors include smell, detected by the olfactory epithelium of the nose; texture, detected through a variety of mechanoreceptors, muscle nerves, etc.; temperature, detected by thermoreceptors; and coolness and hotness through chemesthesis. As taste senses both harmful and beneficial things, all basic tastes are classified as either aversive or appetitive, depending upon the effect the things they sense have on our bodies. Sweetness helps to identify energy-rich foods, while bitterness serves as a warning sign of poisons. Among humans, taste perception begins to fade around 50 years of age because of loss of tongue papillae and a general decrease in saliva production. Also, not all mammals share the same taste senses. Some rodents can taste starch, cats cannot taste sweetness, and several other carnivores have lost the ability to sense up to 4 of their ancestral 5 taste senses. Introduction. History. In the West, Aristotle postulated in c. 350 BCE that the 2 most basic tastes were sweet and bitter. He was one of the first to develop a list of basic tastes. Ayurveda, an ancient Indian healing science, has its own tradition of basic tastes, comprising sweet, salty, sour, pungent, bitter and astringent. Similarly, the ancient Chinese regarded spiciness as a basic taste. Recent discoveries, the receptors for the basic tastes of bitter, sweet and umami have been identified. They are G-protein-coupled receptors. The cells that detect soreness have been identified as a subpopulation that express the protein PKD2L1. The responses are mediated by an influx of protons into the cells, but the receptor for sour is still unknown. The receptor for amyloride-sensitive attractive salty taste in mice has been shown to be a sodium channel. There is some evidence for a 6th taste that senses fatty substances. In 2010, researchers found bitter taste receptors in lung tissue, which cause airways to relax when a bitter substance is encountered. They believe this mechanism is evolutionarily adaptive because it helps clear lung infections, but could also be exploited to treat asthma and chronic obstructive pulmonary disease. Innovation. Taste is brought to the brainstem by 3 different cranial nerves. Facial nerve, specifically the chorda tympani, for the anterior 2/3 of the tongue and soft palate. Glossopharyngeal nerve for the posterior 1/3 of the tongue as well as portions of the upper pharynx. Vagus nerve for the small area on the epiglottis. Basic tastes. For a long period, it was commonly accepted that there is a finite and small number of basic tastes, of which all seemingly complex tastes are ultimately composed. Just as with primary colors, the basic quality of those sensations derives chiefly from the nature of human perception. In this case, the different sorts of tastes the human tongue can identify. As of the early 20th century, physiologists and psychologists believed there were 4 basic tastes: sweetness, soreness, saltiness, and bitterness. At that time, umami was not proposed as a 5th taste, but now a large number of authorities recognize it as the 5th taste. In Asian countries within the sphere of mainly Chinese and Indian cultural influence, pungency had traditionally been considered a 6th basic taste. In 2015, researchers at Purdue University suggested a new basic taste called oleogustus. Sweetness, usually regarded as a pleasurable sensation, is produced by the presence of sugars and a few other substances. Sweetness is often connected to aldehydes and ketones, which contain a carbonyl group. Sweetness is detected by a variety of G-protein coupled receptors coupled to the G-protein gustation found on the taste buds. At least 2 different variants of the sweetness receptors must be activated for the brain to register sweetness. Compounds the brain senses as sweet are thus compounds that can bind with varying bond strength to 2 different sweetness receptors. These receptors are T1R2+3 and T1R3, which account for all sweet sensing in humans and animals. Taste detection thresholds for sweet substances are rated relative to sucrose, which has an index of 1. The average human detection threshold for sucrose is 10 millimoles per liter. For lactose, it is 30 millimoles per liter, with a sweetness index of 0.3, and 5-nitro-2-propoxyaniline 0.002 millimoles per liter. Soreness is the taste that detects acidity. The soreness of substances is rated relative to dilute hydrochloric acid, which has a soreness index of 1. By comparison, tartaric acid has a soreness index of 0.7, citric acid an index of 0.46, and carbonic acid an index of 0.06. Sour taste is detected by a small subset of cells that are distributed across all taste buds in the tongue. Sour taste cells can be identified by expression of the protein PKD2L1, although this gene is not required for sour responses. There is evidence that the protons that are abundant in sour substances can directly enter the sour taste cells. This transfer of positive charge into the cell can itself trigger an electrical response. It has also been proposed that weak acids such as acetic acid, which are not fully dissociated at physiological pH values, can penetrate taste cells and thereby elicit an electrical response. According to this mechanism, intracellular hydrogen ions inhibit potassium channels, which normally function to hyperpolarize the cell. By a combination of direct intake of hydrogen ions and the inhibition of the hyperpolarizing channel, soreness causes the taste cell to fire action potentials and release neurotransmitter. The mechanism by which animals detect sour is still not completely understood. The most common food group that contains naturally sour foods is fruit, such as lemon, grape, orange, tamarind, and sometimes melon. Wine also usually has a sour tinge to its flavor, and if not kept correctly, milk can spoil and develop a sour taste. Children in the US and UK show a greater enjoyment of sour flavors than adults, and sour candy is popular in North America, including crybabies, warheads, lemon drops, shock tarts, and sour versions of Skittles and Starburst. Many of these candies contain citric acid. Saltiness is a taste produced primarily by the presence of sodium ions. Other ions of the alkali metals group also taste salty, but the further from sodium, the less salty the sensation is. The size of lithium and potassium ions most closely resemble those of sodium, and thus the saltiness is most similar. In contrast, rubidium and cesium ions are far larger, so their salty taste differs accordingly. The saltiness of substances is rated relative to sodium chloride, which has an index of 1. Potassium, as potassium chloride, is the principal ingredient in salt substitutes and has a saltiness index of 0.6. Other monovalent cations, e.g., ammonium NH4+, and divalent cations of the alkali earth metal group of the periodic table, e.g., calcium, California 2+, ions generally elicit a bitter rather than a salty taste, even though they, too, can pass directly through ion channels in the tongue, generating an action potential. Bitterness is the most sensitive of the tastes, and many perceive it as unpleasant, sharp, or disagreeable, but it is sometimes desirable and intentionally added via various bittering agents. Common bitter foods and beverages include coffee, unsweetened cocoa, South American mate, bitter gourd, olives, citrus peel. Many plants in the Brassicaceae family, dandelion greens, wild chicory, and escarole. The ethanol in alcoholic beverages tastes bitter, as do the additional bitter ingredients found in some alcoholic beverages including hops in beer and orange and bitters. Quinine is also known for its bitter taste and is found in tonic water. Bitterness is of interest to those who study evolution, as well as various health researchers, since a large number of natural bitter compounds are known to be toxic. The ability to detect bitter-tasting, toxic compounds at low thresholds is considered to provide an important protective function. Plant leaves often contain toxic compounds, yet even amongst leaf-eating primates, there is a tendency to prefer immature leaves, which tend to be higher in protein and lower in fiber and poisons than mature leaves. Amongst humans, various food processing techniques are used worldwide to detoxify otherwise inedible foods and make them palatable. Furthermore, the use of fire, changes in diet, and avoidance of toxins has led to neutral evolution in human bitter sensitivity. This has allowed several loss-of-function mutations that has led to a reduced sensory capacity towards bitterness in humans when compared to other species. The threshold for stimulation of bitter taste by quinine averages a concentration of 8 μm. The taste thresholds of other bitter substances are rated relative to quinine, which is thus given a reference index of 1. The most bitter substance known is the synthetic chemical Denatonium, which has an index of 1000. It is used as an aversive agent that is added to toxic substances to prevent accidental ingestion. This was discovered in 1958 during research on lignocaine, a local anesthetic, by McFarlane Smith of Gorgie, Edinburgh, Scotland. Research has shown that TAS2Rs such as TAS2R38 coupled to the G protein gustation are responsible for the human ability to taste bitter substances. They are identified not only by their ability to taste for certain bitter ligands, but also by the morphology of the receptor itself. The TAS2R family in humans is thought to comprise about 25 different taste receptors, some of which can recognize a wide variety of bitter-tasting compounds. Over 550 bitter-tasting compounds have been identified, of which about 100 have been assigned to one or more specific receptors. Recently it is speculated that the selective constraints on the TAS2R family have been weakened due to the relatively high rate of mutation and pseudogenization. Researchers use two synthetic substances, phenylthiocarbamide and 6N-propylthiouracil, to study the genetics of bitter perception. These two substances taste bitter to some people, but are virtually tasteless to others. Among the tasters, some are so-called supertasters, to whom PTC and PROP are extremely bitter. The variation in sensitivity is determined by two common alleles at the TAS2R38 locus. This genetic variation in the ability to taste a substance has been a source of great interest to those who study genetics. Umami is an appetitive taste and is described as a savory or meaty taste. It can be tasted in cheese and soy sauce, and is also found in many other fermented and aged foods. This taste is also present in tomatoes, grains, and beans. a loanword from Japanese meaning good flavor or good taste. Umami is considered fundamental to many Eastern cuisines, although it was only recently recognized in the West as a basic taste. Umami, or scrumptiousness, was studied and identified by Kikuchi Ikeda, who began to analyze kombu in 1907, attempting to isolate its dashi taste. He isolated a substance he called Ajinomoto, Japanese for at the origin of flavor, later called monosodium glutamate, and sold as a food additive. It produces a strong umami taste. Some umami taste buds respond specifically to glutamate in the same way that sweet ones respond to sugar. Glutamate binds to a variant of G-protein coupled glutamate receptors. See TAS1R1 and TAS1R3 pages for a further explanation of the amino acid taste receptor. Measuring relative tastes. Measuring the degree to which a substance presents one basic taste can be achieved in a subjective way by comparing its taste to a reference substance. Sweetness is subjectively measured by comparing the threshold values, or level at which the presence of a dilute substance can be detected by a human taster, of different sweet substances. Substances are usually measured relative to sucrose, which is usually given an arbitrary index of 1 or 100. Fructose is about 1.4 times sweeter than sucrose. Glucose, a sugar found in honey and vegetables, is about 3/4 as sweet, and lactose, a milk sugar, is 1/2 as sweet. The soreness of a substance can be rated by comparing it to very dilute hydrochloric acid. Relative saltiness can be rated by comparison to a dilute salt solution. Quinine, a bitter medicinal found in tonic water, can be used to subjectively rate the bitterness of a substance. Units of dilute quinine hydrochloride can be used to measure the threshold bitterness concentration. The level at which the presence of a dilute bitter substance can be detected by a human taster of other compounds. More formal chemical analysis, while possible, is difficult. Functional structure. Sweetness. Sweetness is produced by the presence of sugars, some proteins, and a few other substances. It is often connected to aldehydes and ketones, which contain a carbonyl group. Sweetness is detected by a variety of G-protein coupled receptors coupled to a G-protein that acts as an intermediary in the communication between taste bud and brain. Gustation. These receptors are T1R2+3 and T1R3, which account for sweet sensing in humans and other animals. Soreness. Soreness is acidity, and, like salt, it is a taste sensed using ion channels. Hydrogen ion channels detect the concentration of hydronium ions that are formed from acids in water. In addition, the taste receptor PKD2L1 has been found to be involved in tasting sour. Saltiness. Saltiness is a taste produced best by the presence of cations and, like sour, it is tasted using ion channels. Other monovalent cations, e.g., ammonium NH+4, and divalent cations of the alkali earth metal group of the periodic table, e.g., calcium, California 2+. Ions, in general, elicit a bitter rather than a salty taste, even though they, too, can pass directly through ion channels in the tongue. Bitterness research has shown that TAS2Rs such as TAS2R3/8 are responsible for the human ability to taste bitter substances. They are identified not only by their ability to taste certain bitter ligands, but also by the morphology of the receptor itself. Umami. The amino acid glutamic acid is responsible for umami, but some nucleotides can act as complements, enhancing the taste. Glutamic acid binds to a variant of the G-protein coupled receptor, producing an umami taste.